So in the series of biophysical techniques, we will be looking at these two techniques, isothermal and differential calorimetry and atomic spectroscopy. So calorimetry is a quantitative measurement of heat, yeah, and it measures the transfer of energy from one system to another system that has been caused by temperature differences. So usually this calorimetry can be applied in areas that will include the measurements of specific heats of solids and liquids, the heat of vaporization and combustion, and also when we want to determine the rate of heat generation from radionuclides. Yeah. So calorimetry can be divided into isothermal titration and also what we call differential calorimetry. So we look at isothermal titration as the first one. And in this ITC, it is a technique that is used to quantitatively measure a wide variety of biomolecular interactions. That means a direct measurement of heat generated or absorbed when molecules interact. Directly, we can measure the heat that is either being released or absorbed during a biomolecular binding reaction. And simultaneously, now we are able to do a complete thermodynamic profile where we can determine all binding parameters in a single experiment, such as the binding constant, the reaction stoichiometry, and enthalpy, as well as entropy. So the principle of ITC, we have already said it is used to measure the interactions between biomolecules. So it can help us determine the binding affinity, which we normally say it is the strength of binding interaction between a single biomolecule such as a protein, a DNA, and its ligand or binding partner. For example, maybe it's binding to a drug or inhibitor and others. We can also be able to determine the stoichiometry, and this is the determination of proportions in which now the elements and compounds in this biomolecular assay are reacting with each other. In what proportions between the two? How much of the protein is binding to the drug? Then we can also be able to determine entropy, and entropy is the measure of a disorder of a system. And when we are having the transition between solid to liquid to gas through processes such as melting, then vaporization, we are increasing entropy because now the particles from the solid to liquid to gas become more disordered. Yeah. And then we can also be able to determine enthalpy. And enthalpy is the total kinetic and potential energy of a system at constant pressure. And normally we say that a change in enthalpy is the change of heat of a system. Yeah. So now when binding occurs between our two biomolecules, then heat is either absorbed or released. And this is measured by a sensitive calorimeter during gradual titration, because this is isothermal titration. So through gradual titration of the ligand into the sample cell that contains the biomolecule of interest. So let us look at the instrumentation so that now we can understand how the process of titration occurs and the measurement of the changes in heat thereof. So we have the thermocore. So essentially, you can see this diagram here. This is how the setup of the experiment looks like. So we have the thermocore, and in the microcalorimeter, we have two cells. The green one in this case, and this one which is red blue. And one of them contains water, which acts as the reference cell. So the green one has water acting as the reference cell, and now the other one contains your sample of interest. Then these two need to be at the same temperature, yeah, at constant temperature, so that when there is any change, there can be, a quantitative measurement of that. So the heat sensing device detect temperature differences between the cells when there is binding, okay? And of course give feedback to the heaters which compensates for the difference and return the cells again to equal temperature for the next measurement and so on. So when we are making the measurement, the reference cell and the sample cell are set to be at the desired experimental temperature. Then the ligand is loaded here. So this is where you load the ligand. Once you load the ligand, it will, now start ejecting. So the ejection device is inserted into the sample cell containing your protein of interest, so that now a series of small amounts of ligand can be injected into the protein solution, so that we can now monitor the binding between our protein and our ligand in small amounts, that is titrated amounts. So if there is binding of the ligand to the protein, then there will be heat changes, and these heat changes can be detected even a few millions of a degree Celsius, yeah, and then they can be measured and quantified. So this is how the process occurs. So we are having now, this is our sample cell here, and then now we have our injection. We want to now inject the ligand that is in the syringe, and here in our sample cell we have the micromolecule, maybe your protein. So we do a first injection. As the first injection is made, all the ligand is bound to the target, and the signal returns to baseline. So this is, you can see in this graph here. So you have done the first injection, and we are having now it binding the macromolecule ligand complex binding, and then now we have the returning to baseline back in preparation for the next injection. Yeah, so then the injection will continue because we said it is titrated or done in small amounts. So you continue the injection and you can see different spikes here. So this was the first injection, second, third, fourth, and so on and so forth. And as you continue, we're expecting that, of course, the compound or the macromolecule is getting saturated with the ligand, and therefore less continues to bind as we go on, so that also the heat changes will decrease as we go with time. So then a binding curve is then obtained. So you can see this binding curve is obtained. And once it is obtained, you plot the heat changes from each of the injection against the ratio of the ligand and the binding partner in the cell. Once you have developed the curve, now from this particular curve, you can see here, we can now be able to determine the binding affinity. We can be able to determine the change in heat or the enthalpy change here. And then we have the stoichiometry, which we said is now the proportions of binding between the macromolecule and our ligand. So essentially, there is a lot of calculations that will occur to be able to determine now the thermodynamics, yeah, which will include uh, measuring the Gibbs free energy, enthalpy, entropy of binding, etc. So using these relationships, we can be able to obtain the full thermodynamic profile of the binding events between our macromolecule and the ligand of interest. So that is how that form of isothermal titration calorimetry works. Where can it be applied? In drug discovery, yes. When we want to do characterization, optimize candidates, and also check for mechanism of action of this drug in terms of it binding to our target. Then we have quantitative measurements of interaction between any two biomolecules. Just want to know how the proteins interact maybe with RNA, etc. We can also do confirmation of binding and activity. We said that we can be able to determine binding affinity, and then we can also be able to determine the dynamic parameters and stoichiometry. Then we can assess also the effect of molecular structure changes on binding mechanisms. So you have the initial way in which a certain molecular structure binds to a certain ligand. Then you see what if you altered the changes of your molecular structure. Will it affect the binding mechanisms? And of course, analysis of enzyme kinetics when we determine how enzymes bind to substrates. Yeah. The advantages of this is that you can get a basic thermodynamic characterization, getting the stoichiometric constants, association constants, binding enthalpies within a single experiment. Yeah, then we don't need to do labeling with fluorophores or chromophores. We don't need to add other components to help us visualize our results because we depend on heat. Direct determination of binding enthalpy, then interaction in the solution. You can determine what interactions are going on and what changes are they causing in terms of enthalpy, in terms of binding affinities, in terms of heat loss or gain. Then possibility of performing the experiment with optically dense um, solutions or unusual systems such as intact cells, then this becomes very possible. Then you can have this as an advantage because this test is quite fast in terms of determining the binding of macromolecules and their ligands. Disadvantages, heat is universal, so there is likely to be some other heat that is coming from other places. So it's not sometimes it can be a challenge to control any heat and evaluating heat because heat is a universal signal. Large amounts of sample are needed because you need to do titration. Kinetically slow processes may be overlooked. Yes, if we have activities that are kinetically slow, remember this is measuring kinetics, how we are able to bind macromolecules to ligands over time, some sort of reactions such as enzymatic reactions. So if we have binding that is not very kinetically active, then this may be overlooked by this test. A limited range of consistently measured binding affinities can be obtained. So that is also a disadvantage. So these are the advantages and the disadvantages of isothermal titration calorimetry. Now we have also differential scanning calorimetry, which is also thermal analysis because we said calorimetry has everything to do with thermal analysis. So we do a thermal analysis where differences in heat flow into a substance and a reference are measured here as a function of the sample temperature, while both subjected to some sort of control of temperature. So here, the sample and the reference cell are both maintained at a predetermined temperature by the system or the program. Then during the thermal event in the sample, the system will transfer heat to or from the sample. So this is now transfer or flow of heat to or from the sample. Then after it has transferred to or from the sample pan, then we need, so it will transfer heat between the sample or from the sample to maintain the same temperature as the reference and the sample pan. So we start with the same temperature for both, and then if there is any changes because of maybe binding to the sample, then the system will remove heat from that particular sample pan so that it can maintain the original temperature. Yeah. So here we have how the sample setup looks like. So we have a furnace, this one that is U-shaped. Then we have the sample pan and we have the reference pan. Yeah. So what happens is that any differences in heat flow between the sample and the reference is measured with the linear change in sample temperature. So if there is any sample temperature changes here, then there'll be either heat flow outside or heat flow inside so that it balances and gets to the original. Yeah, so those changes can be linearly measured. So we can have one that is within the same chamber, but we can also have those that have been separated. The heaters have been separated. So the principle of this test is that a sample is placed inside the crucible, which is then placed inside the furnace of a differential scanning calorimetry system alongside the reference pan, which is normally empty. Yeah. So by applying a controlled temperature program, isothermal heating or cooling at constant rates, phase changes can be characterized and the specific heat of a material can be determined. Yeah. So here we have the heat flow quantities being calculated based on the calibrated heat flow characteristics of the cell. So we have the two pans, sample and reference pans, and then we have them being put in a crucible, and then we have a furnace controller. Then what will happen is that any heat transfer changes that may occur will be determined and quantified. And now we can understand the various heat flow characteristics of the sample of interest. So now that means that if we understand the heat flow characteristics of a sample, it means that we can classify it as either endothermic or exothermic effect. Yeah, because if it is the sample is going to gain or allow heat flow into itself or absorb energy, then it is endothermic. If it is going to release energy outside, then it is exothermic. Yeah. So that is now how we are able to classify. So we can have common endothermic effects such as melting and sublimation. So if your sample is melting or subliming, then we are expecting that we can have endothermic effects. Sometimes when you have first order and second order phase transitions, evaporation, denaturation of proteins, and galactonization of starch with water events. Then we also have exothermic, maybe when we are having crystallization, oxidation, combustion, fermentation, polymerization, etc. So that way we are able to either check for exothermic or endothermic, whether the sample system was gaining energy or was releasing energy. So now that tells you that this particular test is very important for pharmaceuticals, where we can check for melting points. We can also investigate dissolution of substances in certain solvents. We can check for glass transitions of amorphous materials. We can check for heat transitions such as melting and crystallization. We can determine purity because if there is contamination, then the melting and also boiling points can be altered. Crystalline amorphous phase quantification, polymorphic transitions, we have processing conditions, we have we can check for compatibility interactions between components, and we can also check for kinetics when we have decomposition of compounds, chemical and thermal stability. As they're looking for chemical and thermal stability, so they decompose, so you can check for decomposition kinetics. So now this is atomic spectroscopy, and we have two: absorption spectroscopy and emission spectroscopy. I mentioned a bit when we looked at what it means to leave ground state and get excited to excited state on a higher energy level, and then what it happens when we are having emissions. Yeah. So now absorption spectroscopy is now when we have after an irradiation, we have the transition from a lower energy level to a higher energy level. Then emission spectroscopy is now when coming from a higher energy level, releasing some radiation, then goes back to a lower energy level. So absorption spectroscopy can be atomic spectroscopy, that is, can be divided into absorption spectroscopy, AAS, can also be divided into mass spectroscopy and also emission spectroscopy. So we are going to look at these two, and we'll also look at mass spectroscopy in a later lesson. Yeah? So atomic absorption spectroscopy, AAS, is an analytical technique that measures the concentrations of elements, and it makes use of the absorption of light by these elements in order to measure their concentration. So the atomic absorption spectroscopy will quantify or give us the concentration of the absorption of ground state atoms in gaseous state. So the atoms absorb UV or visible light, and once they have absorbed, they make a transition to higher electronic energy levels. Then the analyte concentration is then determined from the amount, because all of them will absorb differently. How an element is going to absorb UV or visible light will be different. So depending on the amount of absorption, then we give the concentration of that analyte. So concentration measurements are usually determined from the working curve after calibrating the instrument with known standards. Standards are important. So already we have concentrations that have been known before. So we use those to help us determine the concentration of our analyte of choice. So in this AAS, it is very common technique for detecting metals and metalloids in environmental samples. So if you are doing an investigation of environmental sample like water from dams, maybe ocean, etc., you can be able to determine metals or metalloids, such as the ones that I have reminded us on this table, this periodic table. You can be able to look for them. The high ones that have been highlighted in pink. So those metals and metalloids is good method to use AAS to determine their concentration. So how does it work? It has four elements. The light source, yes, we said we must put a light source. Then we have the atom cell, or where we are having our sample. Monochromator is now for separating light, so that now during the detection and the readout, we can now be able to map what is our absorption and how much concentration does it have. So the light source is usually a hollow cathode lamp, like the one that you can see here. It's a hollow cathode lamp. Yeah. Then it contains a tungsten anode here. And now the hollow cylindrical cathode. So the cylinder here is the cathode. Yeah. And these are sealed in a glass tube. So you can see now the outside shell, which is quartz window, it's glass that is now there. The glass tube that seals with inert gas. Then each element has its own unique lamp which can be used for the analysis. So this is now when you're looking from the top, the anode, and then the cathode tube. So it works by applying a potential difference between the anode and the cathode, yeah? So that the metal, the ionization of the gas atoms occurs. Yeah. Once the gaseous ions are being ionized, the gaseous ions bombard the cathode and eject metal atoms from the cathode in the process called sputtering. So what happens is that this cathode will be made up of the metal in which you want to do investigation. Then we already said that we will ionize some gas because of the difference between the cathode and the anode, the potential difference that has been created. So once these gaseous ions have been formed, they bombard or hit the cathode and release or eject metal atoms in a process called sputtering. So this is how it looks like actually. So we have the anode and we have the cathode. So you apply power and there'll be a potential difference. So the shape of the cathode is hollow cylindrical to concentrate the emitted radiation into a beam so that it can pass through a quartz window and all the way to the vaporized sample. So since the atoms of different elements absorb characteristic wavelengths of light differently, analyzing a sample to see if it contains a particular element means using light from that element. So for example, with the lead, a lamp containing lead emits light from excited lead atoms that produce the right mix of wavelengths of now that can now be absorbed by the lead atoms from the sample. So, a beam of electromagnetic radiation emitted from excited lead atoms is now going to be passed through the vaporized sample, and some of the radiation will be absorbed by the lead atoms in the sample. And if we have a great number of atoms there in the vapor, there will be more radiation that will be absorbed. Yeah. So in the atomizer, so we have the light, radiation, and then now we have an atomizer. So what happens is that the elements to be analyzed need to be in atomic state. So atomization is now the process of separating particles into individual molecules so that you break them apart into atoms. And this way is done by exposing the analyte to very high temperatures in a furnace or flame. And the role of this atomic cell is to dissolvate the liquid sample, and then the solid particles will be vaporized into becoming gaseous. Yeah. And once they become gaseous, the atoms will now be available to absorb any radiation that is being emitted from the light source. And after now that, they can be able to give us a measurable signal of the concentration. So sometimes you can use a flame. So the flame atomizers, uh, you can use them. For example, you can see this atomization technique here where you have a flame, and this flame can be, you know, maybe having fuel of oxidants such as gas and air, hydrogen and air, and other acetylene, and we also have other organic compounds combined with oxygen, combined with nitrogen oxide, etc. So they can see that the temperatures vary and they are quite high, and this is degrees Celsius. So flame atomizers can be used in all atomic spectrophotomic spectroscopic techniques, and they convert analyte into free atoms. So whatever you want to analyze, make it into vapor phase. Then of course heat is required, and the routes for sample introduction are important. So this is when you use a flame. Graphite furnace can also be used, and it is better because it can be able to accept solutions, salaries, and also solid samples. And also because it's much more efficient in atomization than flame, and it can directly accept very small absolute quantities of sample. So it also provides a reducing environment for easily oxidized elements. Samples are placed directly into the graphite furnace, and furnace is electrically heated. So you place your sample onto a graphite furnace, and then the furnace is electrically heated in several steps to dry that sample, then convert it into ash organic matter and vaporize it. Yeah, and it can accommodate very small samples. That because the high energy is provided, and it can be able to now excite and atomize those particles. So essentially, we have said that we already gotten our light source. Now, in atomizer is where you have your sample of interest, and it must be made into gaseous form. Then now after that we have now the monochromators. So the monochromator is a very important part of this particular technique because it is used to separate all the 1,000 different lines of radiation so that it makes it into one. It selects a specific wavelength of light which has been absorbed by the sample and exclude all the other wavelengths that have not been absorbed by the sample, so that only this is directed to the readout or the detector. So now at the detector, the light from the monochromator is now directed, and it is directed to what we call a photomultiplier tube. This function is to convert now a light signal into an electrical signal, and this now will help us to be able to map the light intensity. So this electrical signal is fulfilled by a signal amplifier, and the signal can be displayed now on the readout. And now we can have now the data printed as requested to now see what was the concentration of our desired analyte. Now this particular process can also have interferences that could affect its function, and some of these interferences are absorption of source radiation. So the element other than the one of interest may actually absorb. So maybe you're having lead as your element, but maybe there is some aspects of iron in the whole cascade. So that might interfere with your results. Ionization, the formation of ions rather than atoms causes lower absorption of radiation. So maybe as you're doing atomization, we have, instead of making them into atoms, they now become ionized instead, and that may reduce the amount of absorption of radiation that may occur, hence giving us uh, less concentration as it should be. Then self-absorption, so the atoms of the same kind that are absorbing radiation will absorb more at the center of the line than at the ends, yeah, and that's resulting into the change of shape of the intensity. So we can have self-absorption because atoms also have different sections between the edges and the center. So that self-absorption will give us incorrect intensity. We can also have background absorption of source radiation, so that we have presence of a particle from an incomplete atomization. This problem can be overcome by making sure that the flame is very good temperature, or use graphite furnace to ensure that you have complete atomization. Then transport interference, the rate of aspiration, nebulization, and transport of this sample throughout the system may also interfere because of sometimes different viscosity, surface tension, vapor pressure, and density. So the last one is atomic emission. So that the one was relying on atomic absorption. Now here it's emission. So the emission spectroscopy is an analytical technique that is used to measure the concentration of sample, and it uses quantitative measurements of the emission. The amount of emission will give us the analyte concentration. So the analyte atoms are promoted, of course, to a higher energy level by sufficient energy that is provided by high temperature of atomization sources. So, of course, the first thing is to make sure that the analyte atoms have been promoted from a lower energy level to a higher energy level. Yeah. So, after that, we have now the excited atoms decaying back. They will decay back. Yeah. So, as they decay back to the lower energy levels, they will emit some light. So that light that is emitted is now the one that will be passed through a monochromator or filter, and then it is detected by a photomultiplier tube. So the instrumentation of atomic emission spectroscopy is the same as that of the atomic absorption, but without the presence of a radiation source. So remember, in this case, we don't have a radiation source. So in absorption, we were radiating, so that after we have done radiation, we can have the atoms that have been already atomized absorbing the radiation, and through absorbing and now coming from a low energy level to a high energy level, we measure the concentration of our analyte. Now, in emission, we usually don't have a radiation source. Yeah. So here we have excitation source to excite from a lower energy level to a higher one, and then measure the decay or the emission when we are having now the return to a lower energy level. So this technique is also known as the optical emission spectroscopy, and it helps us to study the radiation emitted by excited atoms and monoatomic ions. So the relaxation of atoms in excited state results into emission of light. That's what we mentioned, and produces a line spectra in the UV visible and the vacuum UV regions that can now be quantified. So this is used for qualitative identification of elements present in the sample, and We can also be able to measure the quantitative analysis. We can also have multi-element techniques where we can, determine many different elements at the same time. Can be used to determine metals, metalloids, and some non-metals simultaneously. So here we have an excitation source to excite the atoms from lower energy level to higher energy level. So that then after that, whatever light is emitted as their decay can be passed through a filter and then detected. So the sources of excitation can be also flame. We also have what we call alternating current. We have DC, direct current. We have alternating current spark. We have direct current plasma, microwave induced plasma, and we have inductively coupled plasma. So of course flame, we normally use flames such as hydrogen, and we have acetylene. Then when we come to direct current, it is generated with a potential gradient of 50 to 300 volts. Yeah. Then for alternating current, we normally employ a potential difference of 1,000 volts or more. Then we have alternating current spark, where we do higher voltage compared to just the alternating current. Here we increase, that is 10 to 50 kilo voltages. Yeah. And then we have what we call plasma. So this is an electrically conducting gaseous mixture containing significant concentration of cations and electrons. And these ones are the ones that can be divided into inductively coupled plasma, indirect current plasma, and microwave-induced plasma. So it's a gaseous mixture that can also help us to excite our atoms so that they can go to a higher energy level. Then we can determine the emission of light when we have decay back to the lower energy level. Now, this part here just tells us some of the techniques that you will have based on the excitation source. That based on which source you use, we can either now have the technique being called flame photometry, we can have furnace or electrical excitation, and we can have inductively coupled plasma technique. So this is an example, the direct current plasma technique. So the direct current plasma is created by electronic release of two electrodes. Yeah. So the samples are going to be placed between two electrodes. So the distance between two electrodes sputters the sample atoms. Yeah. So that now they can be encouraged to emit, yeah, to emit the light that is now going to be useful in determining their identity as well as their concentration. Then inductively coupled plasma. So plasma generated in a device that is called torch. So what happens in this one is that you pass your sample. You can see your sample down here. Your sample aerosol or vapor in argon. So you pass it in between, and then what happens is that we have radio frequency induction coils surrounding. So they generate up to 2 kilowatts. Yeah. So this radio frequency is very important. And then we also have what we call the tensor coil producing initiation spark. Yeah. So the ions and the electrons interact with the magnetic field and begin to flow in a circular motion. So the ions now from the sample will now begin to flow in a circular motion. So the resistant movement or collisions of now the electrons and the cations with the ambient gas that is in this system leads to some sort of heating, yeah, so that the sample introduction is analogous to the atomic absorption. So Once now that happens, you can see that now the sample ions and electrons are moving within the magnetic field. So they get now excited, and through that excitation, we have the emission, and the emission now can then be determined, and it will give us the identity of what sample was flowing into these quartz tubes and how much of it is there. Now, we have different ways in which we introduce the sample. Like here, for example, here, how do we introduce this sample here or here? Yeah. So one of them is using nebulizer, and a nebulizer usually converts a solution into a fine spray or aerosol. Yeah. So we pass our solution. So this is our sample solution. It passes, then it mixes with gas so that it is made into an aerosol, and now you can inject it into the required technique. So we have two ultrasonic nebulizer that uses ultrasound waves to boil the solution, and we have pneumatic nebulizer which uses high pressure to entrain the solution into gaseous form. Then we have what we call electrothermal vaporizer. This is the electrothermal vaporizer that now uses electric current to rapidly heat a crucible, and this crucible contains our sample. So the sample now is carried to the atomizer by gas, and only for introduction and not for atomization. So this can help us now to introduce our sample into the technique that we want to use. So the sample holders, for us to be able to, introduce the sample into the electrical discharge, you can have those that carry solid samples and of course those that are carrying liquid samples. So we have looked at what is the excitation source. We talked about many different direct current, alternating current. We looked at flame, etc. And now we have also seen how do you inject your sample into the system. Now we do also have monochromators. After now light has been emitted through this system, we now need to filter the wavelength so that only the wavelength of the material being analyzed is directed to the detector or photomultiplier tubes. So once we have had the filtration, we have the photomultiplier tubes and also photographic plates that are now our detecting mechanism. So for quantitative analysis, photographic plate is used, yeah, on which all the emission lines from the samples can be recorded. So then this photograph of emission spectrum helps us in the measurement of the wavelength of the radiation lines. Now from these lines, emitting elements can be identified. Then the instruments with photographic recording is usually called a spectrograph, and the one that uses photoelectric device is called a spectrometer. Now we talked about the photomultiplier tube in the previous one that converts light into electrical signals that can now be showed using a readout device. So we have advantages of either using photographic plate or photomultiplier. Photographic can be synonymous to what we get when we do X-rays. Yeah. So normally the advantages of photographic plates and photomultiplier tubes, we have larger number of spectral lines can be recorded when you use photographic, and then photographic also provides a permanent record as compared to just having a readout on a screen. Then emission spectrum can be integrated by a photographic emulsion for a long time. Yes. And then these emulsions are preferred because of their high sensitivity to UV and visible regions. But these photographic plates also do not show quick response to spectral lines, and the interpretation is also not very easy because a photomultiplier tube, once it gives us a readout, it can also be able to do the interpretation for you. But photographic, you would need to analyze it yourself visually. Then in photographic detection, controlled photographic development is required, and this requires much time and increases the risk of error. So I have just put an example of some of the atomic emission spectroscopy instrument types that exist, such as we have what we call sequential scanning and slew scanning machines or instruments. We have a multi-channel that is able to measure intensities of large number of elements at the same time, about 50 to 60. And we also have Fourier transform. I'm sure you probably have had Fourier transform, or you may come across it in another technique. Now, what is important about all the emission spectrometers that we have, what is important is that they should have high resolution. We should have rapid signal acquisition. They should have low stray light, like light should really fall on the monochromata so that it does not, fall away. Otherwise, if it falls away, then it will give us reduced intensity at the end. A wide dynamic range to also be able to determine different elements, whether multiplex or not, but should be able to detect at least a wide range of elements, should be accurate and precise in terms of wavelength identification, should have precise intensity readings so that we can give the correct intensity and concentration, should have high stability with respect to environmental changes, yes. Easy background correction so that you can remove any background noise, and if possible, have them computerized so that we can easily get a readout, have manipulation and interpretation, and also store our data for future reference. What are the applications of atomic electron spectroscopy? One is qualitative analysis, because we can be able to determine the elements that are in an analyte. We can also be able to do quantitative analysis, where we determine the intensity or how much of the element is in the analyte. And we can be able to detect a number of elements. For example, approximately 60 of them have been standardized so that we can be able to identify their presence and also quantify them in samples. And this can also be applied in determining the impurities of things like copper, aluminium in iron and steel in metallurgical processes, so that the percentage that is determined is 0.001% in iron to 30 in steel. So sometimes when you are making these particular structures, would need to determine what is the percentage of iron, what is the percentage of iron in steel, and we can now be able also to determine, do we have any other impurities in that particular structure. We can do what we call lubricating oils can be analyzed for certain elements so that we can make them safe. Then we have solid samples, and animal tissues can be analyzed for several elements such as potassium, zinc, etc. Then to detect 40 elements in plants and soils, thus metal deficiency in plants and soils can be diagnosed. What are the advantages of AES? It's highly specific, sensitive, you can determine or identify metalloids. The sample can either be in solid or liquid state. The technique requires not so much sample preparation, and we also don't need any treatment of the sample before it can be used. And the spectra can be taken simultaneously for more than 2 elements without separating them. Then we have also rapid results, particularly if it is automated. It can take 30 to 60 seconds. Disadvantages is that, for example, if you're using Fourier technique, the equipment is quite costly. Yeah, then the recording is done on photographic plate, which can take time to develop and print if you're not having the automated system. Then spectrograph is essentially a comparator. For quantitative analysis, standards of similar compositions may be required. Then the radiation intensity are not always reproducible, especially when you repeat the tests. Then the method is limited to analysis of elements. Here I've done a table that compares both the atomic absorption and atomic emission spectroscopy. So in absorption, we said we can trace um, the amounts of metal concentration in complex matrices, and even in emission, you can be able to check for metal concentration in matrices. In AAS, it depends upon the number of ground state atoms, but in AES, it depends upon the number of excited atoms. So here it was about ground state atoms, and then atomize them, but here we are looking at the number of excited atoms. Then therefore absorption, it measures the radiation absorbed by ground state. Emission measures the radiation emitted by excited atoms. Then the presence of a light source in absorption is apparent, and here we don't have a light source, we have an excitation source. Then in absorption, the temperature is in the atomizer is adjusted to atomize the analyte atoms in ground state only, while the temperature in the atomizer is big enough to atomize the analyte atoms and excite them to a higher energy level, so that now we can measure when they decay how much radiation do they emit. Yeah, that is it. You can channel any questions on my email and I will be happy to answer them.